My name is Dr. Chris Jenkins, and I am the CEO of the Orianne Society and the host of the Snake Talk podcast, the podcast where you learn about nature's most feared, maligned, and persecuted animals. I invite you to listen to this conversation, and maybe you'll find that what you perceive as fear is actually rooted in a deep fascination. Welcome to the Snake Talk podcast. I am sitting here with Lauren Fuchs, who is a Ph.D. candidate at George Mason University. And this is actually the first time I've ever spoken to Lauren, but I was made aware of her work because here at the Orianne Society, we have a small grant program where we give out small amounts of funding to graduate students who are working, in particular in the southeastern part of the U.S. and sometimes a little bit broader. And Lauren was one of the students selected to receive one of those grants. And so I want to start especially the ones that are kind of snake focused in the future. I want to start highlighting a lot of those projects because they're all pretty interesting. And I think you all would be interested in learning about them. So as I mentioned, Lauren's a graduate student and she has a really kind of interesting project, and it revolves around snake fungal disease, but it's getting beyond where we've been for a long time with snake fungal disease, meaning just kind of documenting presence, around the country or beyond. But, it's starting to think about, causes, impacts, how animals can defend themselves from this. So we're going to talk a little bit about snake fungal disease and her research and some of the interesting things she's been working on of late. So Lauren, welcome to the podcast. How are you doing today?
I'm doing well. How are you?
I'm doing great. Thank you very much. So if you've ever heard an episode, you know that I like to get to know my guests a little bit before we get into the meat of the podcast. And we're going to do that here. And I like to, I always kind of say that snakes are, for most people in the world, an interest in snakes or even reptiles and amphibians is a little bit of kind of an eccentric outside of the box interest. And so I always like to hear how people ended up there. And so where did that begin for you? Is this your interest in snakes? Is that something you found as a child or something you found later in life?
No, much later in life, actually. My family was never outdoorsy. I've never really seen too many wild animals outside of squirrels and everything that's in your backyard. And I would probably, I didn't really come across many wild snakes when I was younger, but probably would have run away because I didn't know much about them. We never went camping, never went hiking. It was actually when I was kind of changing careers, I went back to school and was interested in going into, teaching in biology. And I took a biology class and then kind of followed an instructor into a zoology class. And we went on a field trip and I saw the interaction with all these animals, and it was so interesting to me. I was just kind of in awe of what was out there in nature, and just slowly became more and more integrated into, what was out there and being outside. And I actually started getting involved in very, small, simple research, or I guess some projects like cull surveys. And then I started swabbing frogs to help out with another project. And then I started, seeing snakes and holding snakes and was just finding it so fun and thought they were such an amazing animal. And it just grew from there. So I ended up for my master's, I kind of weaved from out of, frog BD research and then moved into OO research and snakes. And my fondness for them has just continued to grow. I even have a pet snake at home, a ball python. So a lot of people, especially in my family, are just shocked at the direction I'm going on, and for sure.
So let's back up a little bit. So you're a child, and it sounds like, did you did you grow up in kind of like a more of like an urban setting? It sounds like you didn't have much connection to the outside growing up. And so first question there is kind of where did you grow up? What kind of setting? And then how, when you went away to college, it sounds like you found, as you mentioned, biology and took some zoology classes. What was it, even if it wasn't snakes, what was it, with kind of that lack of background with nature and science, like what was it that prompted you to want to study biology as an undergraduate?
I've had a very circuitous path as far as my education went. I grew up in Long Island, New York, so suburbia. I only learned recently that my parents did see snakes in the yard growing up. I had never seen them. But yeah, we were, a pretty indoor family. I went away to college. I went to GW in Washington, D.C. So there are snakes out and about here and there, but yeah, not really an outdoorsy area. I did round one of school and kind of went back to school with more of a human anatomy interest. So I had to kind of retake some of my biology classes. And then I don't know, just a few things that popped out in biology and kind of expanding my horizons with, what there is to learn in biology. It's not just all about humans. Just slowly started to spark my interest. I'm like, oh, there's other things out there, and you can enjoy biology, but it can expand, far and beyond just the human body.
Mm-hmm. And you mentioned you went back to school. So did you have like a career before biology, or did you study something else before biology?
I did. So, undergrad, I did psychology, and then I went and ended up doing a first graduate degree in exercise science, and then got a related job for about 4+ years based off of that degree. And as I was doing that, I started taking classes again, realizing I didn't feel fulfilled. I wanted to teach, and I enjoyed science. I enjoyed biology, and I wasn't getting to use that at all as much as I wanted, at least.
Yeah. Well, that's a great message for the audience. Whatever it is you want to do in life, it's never too late. You can always go back and retrain for something else. So, okay. So you're doing your undergrad, and where was your undergrad? Was it at George
Washington University in DC?
In DC. And then you kind of in biology and start getting more interested in zoology. And you finished that degree, and then you mentioned that you did a master's working with frogs. Was that, or with amphibians? I don't remember if it was frogs. And then, but was that also at George Washington, or where was that?
No. So actually, it was when I went back to school is when I first started getting interested in, expanding in science. But I went to a community college in Virginia, Northern Virginia Community College. Okay. To start picking out a few of the classes that, trying to find my passion. I'm not going to go back for a full program. And it was at community college where I took those classes that inspired me. Gotcha. And then from there, I applied to the graduate program for a master's at George Mason. And then I'm continuing my next level of the degree at George Mason as well. Very confusing. I know there's a lot of steps.
Gotcha. So you didn't pursue a second bachelor's. You went and got you took some classes and then did the master's. And so, let's talk a little bit about your master's research. So you were at, did you say George Mason or George Madison?
My first master's was GW. My second one was George Mason.
George Mason.
Attempt number 2. Yeah.
And so let's talk about a little bit about that. How did you end up deciding to go to that school and work on that project?
I started doing research in this area. It wasn't really affiliated with my own particular project, but actually that kind of grew. And from doing some sampling and stuff, we decided to build a bigger project out of it. And I just felt like I had a team in this area that I had been working with, a lot of I guess now colleagues that I did some research with at the community college who I had met along the way and who inspired me and taught me all I eventually learned about, this area of science. And I felt like I had a lot of support in the area, friends and then, of course, the colleagues and professors and things like that. So I thought this was a good place to stay. And I knew a lot of people that had gone to George Mason, and it felt like a comfortable place.
Yeah, great. And so you mentioned that you were working on amphibians. Tell us a little bit about the project, your master's thesis.
Yeah, so I did kind of two intertwined projects. The first one was just simply looking, pretty much at prevalence data of BD at two sites. Actually, one was in Virginia, one was in Maryland.
Sorry, what I know, but what is BD for our listeners?
Oh, I'm sorry.
No, that's okay.
Batrachochytrium dendrobatidis. So it's chytrid causes the chytrid fungus, essentially what causes BD. And so my first experience with a herpetology disease. So I made my whole project out of herpetology diseases. And then I looked a little bit at ranavirus to see if there are any sorts of, co-infection occurring. We didn't get very good data out of that project, probably because of our sampling, but it was still fun to explore that as well.
Were you looking at prevalence in the region of the university, or was it somewhere else in the US or beyond?
We looked at two different sites. One was like a very heavily residential area on with a big wetland in the middle of it in Fairfax, Virginia. And then the other one was a environmental research center out in Annapolis. So it was interesting to kind of compare and contrast, the results from there. So we at least had two sites, but with, a bunch of species. So we got to make some, interpretations of the results.
So your interest, and not to give too much away, we're going to get into this when we talk about your research, but your interest in kind of like landscape or disturbance effects on disease in amphibians and reptiles started then. It isn't a more recent thing. Interesting. So what did you find in your thesis?
We had relatively high prevalence rates. Overall, a relatively high prevalence rate. I believe it was in the 30s. I don't remember off the top of my head exactly, but compared to other studies, ours was quite high. We did not separate the sites at that point because our sample size was pretty low. So we kind of collected all the data together, and that gave us an opportunity to really look more across the species level and didn't give us too much information about, site or landscape information. But I was still curious.
Yeah, gotcha. Okay. Last question about that, just speaking generally, in that part, so this is Northern Virginia and I guess Eastern or maybe more Central Maryland, but are there certain amphibian species that are known to have really, say, declined from BD over time, or is that something we don't know?
I haven't been super up to date on the BD literature. I don't know in this area as much as kind of the local impacts, and I think it was, if I remember off the top of my head correctly, it was the bullfrogs that seemed to be having the highest prevalence levels. I know obviously in other parts of the world, there are associated declines and there are species that are far more impacted than species around here. But there have been, some BD and I think RV related occurrences where, there were population level impacts.
Yeah. So how does the chytrid fungus, or how does it impact an amphibian? Like, what is it doing to the amphibian that kills it? It's
keratinophilic, which is similar to the OO pathogen, which I'll talk about shortly. But yeah, it feeds on the skin. And so kind of this was more or less the inspiration for my current work because, if it's affecting the skin, what is different about the skin that makes, certain species deal with infection better than others? If it's feeding off layers of skin, obviously that's going to be a significant problem for the amphibian who does a lot with their skin. But they have done a lot of research where, certain amphibians have certain fungus and bacteria on their skin that actually fight away, I'll put that in simple terms, fight off infections from pathogens better than others. And there's been a lot of research on that. And so I think that needs to be expanded.
Yeah, Great. Well, we'll get into that here in a few minutes. Let's talk about your PhD. And I'm not, the bulk of our conversation is going to be kind of around your PhD research. So we don't need to talk about the research side yet, but I'm just curious how you ended up. You're at the same institution, you're working in the same general area, looking at disease impacts in herpetology, but you've moved now from amphibians to reptiles. And so I'm just curious kind of, along the lines of, your story of where, how you got to where you're sitting today. How did that all happen?
Well, a big reason I kind of stayed in the area, aside from being, I guess, relatively still close to friends and family, which was important to me. So I didn't want to make a big move, although I did have an opportunity to travel across the country and do some my studies there. Like I said, I have a network here that I'm comfortable with. I'm comfortable with the area. I'm comfortable with a lot of the research sites. And the reason I decided to pursue the next level up the degree is because my plan is to, my career plan is teaching. I'm currently a professor and I have a master's degree, and I would like to get the next level up. And that opens up a lot of doors for me as, in teaching.
Gotcha. So you finished your master's and then you started your career. Essentially, you got your job, and you're doing your PhD to kind of not necessarily to go find a new job, but to bolster what you're already doing. And so tell me about, I guess, first then getting this job. How did you end up? Where do you work? How did you end up getting this job, and how did that all play out?
I guess kind of a funny story is the college that I went to take classes at for my career change is now where I work. I knew I wanted to go into kind of the teaching world, and I didn't really understand the difference between different kind of colleges and universities, and the idea of, having to focus on research or teaching. And so when I went to community college, I was really inspired by, the atmosphere, the care of the professors, the level of involvement for students and the opportunities. They're so much different because I know I'm in a weird place, and I've gone to multiple types of schools at this point. I've experienced a lot of, university life and college life. And I realized that that's where I wanted to teach. And so I started adjuncting because I had the older master's degree, which allowed me to start teaching, had the credentials for it. So I started teaching intro biology classes as an adjunct as I did my master's. And I was persistent and kept applying for a full-time job at the university. And I don't know, they finally, I guess, gave in and gave me a spot. And I think a big part of that is actually during the time I was adjuncting, I also worked really closely with a lot of the faculty. And we started kind of an informal research program, and we were able to get a lot of students involved in, collecting data and doing the lab work and things like that. And they actually ended up on some papers as well. And so this is something that, the faculty that, I worked with, my colleagues and I are all trying to improve at the community college level. And so it's a big reason I wanted to stay, because I had that opportunity. I don't have to focus on research, but I can do it still, even past school. I can, bring research opportunities to the students and teach them, the same way I was given the opportunity that changed my mind, to go out and experience research. I want to do the same for other students. So
that's great. So you're teaching what types of classes do you teach? You're still teaching intro bio, or do you have kind of a whole variety of things you teach these days?
Yeah, currently, while I get through my PhD, I'm keeping it as to, the level I'm most comfortable with. So 101 and 102 are the two classes that I've been teaching for, 7+ years now. But I do hope once I'm done with the PhD and I have a little more time to, create, courses and work on those, definitely going to want to expand to environmental and zoology and classes like that.
Great. So you're teaching, you've got your job. It sounds like it's the job that, you at least plan on working for a while. And, but you decide that getting a PhD will help. And so you you start a PhD program at the same institution where you did your master's. How did you find this amazing grant program? No, but how did you find? So, the connection that we had was a small grant program, and we provided a small amount of funding to help with your research. How did you end up finding that, and how did you find the process, applying and all of that?
Well, first, I am part of a lot of these, programs. I'm a member of these societies and things, so I get constant emails on these opportunities. And in addition to that, obviously, part of being a graduate student is looking for grant opportunities. And it's obviously not super easy to come by when you don't work with a super cute, fluffy animal. It's a little bit of a niche field to ask for money for. And, the people that care about them are the people that are, studying herpetology and appreciate the amphibians and reptiles. And so that's, the area that I looked at. I said, who cares most about what students are doing to become involved and to improve, the conservation of amphibians and reptiles. And that leads me to, a variety of, groups. Many of them are centered in the Eastern US, which is, pretty much how I found you and, the Virginia Herpetological Society, also SSAR, all of them.
Okay, great. Well, we're glad to have you. We're glad you applied. Let's get into kind of the core of this episode and talk about what is the kind of the central focus of your research, which, to summarize, is kind of looking at how, this particular disease, OO, as you called it earlier, or snake fungal disease, as a lot of people know it as, how the microbiome of a snake, affects its immunity relative to SFD, and then how different potential landscape scenarios, levels of disturbance would potentially impact that. But we'll kind of build on that through the course of the episode. And I want to just start with SFD. And if people want just a really in-depth discussion of SFD, we did an entire episode with Matt Allender. People can go find that. But I do want to spend a couple minutes talking about it so people really understand the disease who haven't seen the episode. So first of all, what is OO? What is snake fungal disease? How would you describe it? Just give us the 100 level description of it, the 101. Snakes are one of the most persecuted groups of animals in the world, but we are dedicated to changing that. If you like what we've been discussing here on the Snake Talk podcast, here's an opportunity to turn that inspiration into action. Head over to www.orianne.org and explore how you can support the Orianne Society. Click on Take Action on our menu to make a difference.
Sure. So I guess OO is the abbreviation for Ophidiomyces ophidiocola. And they only recently changed the I and the O addition in there, so it gets pronounced differently sometimes. It is a keratophilic fungus, so it feeds on keratin, like what makes up skin. And it can, so even if it infects the skin or is on the skin, it doesn't necessarily mean it leads to a diseased state, which is known as ophidiomycosis, or more formally known as snake fungal disease. And snake fungal disease is impacting a very wide range of snake species, not just across the United States, but across the world, across most ecoregions of the world at this point. And it has a huge, very variable effects on snakes, which is probably the most interesting part to me, because of these differences that we see, but very typical signs or clinical manifestation that, as it's often said, is, crusty scales, nodules, lesions that are often impacting, things like sensory organs around the mouth, the cloaca, the eyes. In some cases, you can have what's called disseminated ophidiomycosis, which can become kind of a systemic internalized infection, damaging many of the internal organs. it's still, unclear if there are population level effects really occurring at this point. But it does seem to have, like I said, variable impacts. Some can get this infection, some can get some crusty scales here or there. Some will have, severe clinical manifestations. Some will clear it in, a couple days out in the sun, and some will, face mortality. And we don't thoroughly understand why these differences are occurring.
Yeah. We do know it has had some population level impacts on some snake populations that are relatively small, maybe inbred in some cases and isolated. Right. But yeah, more broadly, we don't know. And as we've talked about multiple times, like we've done a lot of prevalence work in the Southeast. And I mean, it's an incredible number of species. Eastern indigo snakes, which is one of the main ones we work on. I mean, like the larger percentage of Eastern indigo snakes have this. So where did this come from? Again, people can get more in-depth discussion, but I'm just curious, is this a new thing that's spreading across North America or across the world? Is this something that's always been here? What's the latest on what we think about the origins of SFD?
So there's kind of some competing views on, how and when this showed up. There's like the endemic hypothesis that it's been here for a very long time, or that it's novel and recently appeared. And it seems there needs to be more research on this for sure, especially, as far as the, understanding the genetics of the pathogen itself will help us understand it. But it seems that it has been here for a while only because we have, some samples collected back from, I think they found it as far back as 1945 in a museum specimen that it's been present. But there's support for both sides, whether it has been here for a long time, whether it's endemic or whether it's novel. Some are supporting the idea that it's just been introduced and reintroduced on many different occasions over time. And so I think there's more research needed there to determine the history of it.
If it was introduced and reintroduced, as you mentioned, do we know what the origin is? Is it a certain geographic region? Is it a certain aspect of, say, the pet trade? Is it do we have any idea if it was introduced, what that kind of vector might have been?
So I'm not, entirely super knowledgeable about the genetics of the pathogen, although this is one aspect that I am looking to study a little bit going further in this project. I have this written down somewhere, and I can't remember off the top of my head the exact, ideas and thoughts as far as, whether if it is endemic versus novel, like where it would have come from. But I know that there are 3 clades, and there are some big distinctions between the clade that's in Europe versus the clade that's in, North America. And then the clade that includes a lot of captive snakes. So there are a lot of differences. So it's hard to really pinpoint a source, but they're looking at, whether there are a lot of recombinants present or not, which can, tell you that it's been here a long time and just evolving, or if there's just a sudden change in the genetics that shows you that it kind of just popped up here. Yeah. So there has been some research into the genetics, the population genetics of the pathogen itself, but definitely an area that needs a lot more work.
Gotcha. Okay. So we talked about, we did mention it, but in passing, the idea there's a lot of variation in prevalence. And when I say that, I mean things such as different species, like prevalence can be much, much different across, suite of species that live in a given area. Certainly we've seen that down here in the Southeast. And I think you've seen some of that up in that mid-Atlantic region as well. And then even within a species, I don't know about prevalence, but if, I already mentioned, in particular with timber rattlesnakes and with Massasaugas, there are examples of kind of isolated, small, maybe genetically, inbred populations actually being impacted pretty heavily by this. So even within a species, I don't know if it's prevalence, but certainly the impact looks like there's variation around that. So there's variation in how this disease is impacting how it's distributing itself amongst species and so on. And so what I'm getting at is the factors that kind of dictate that, and your research revolves a lot around that. In one of them, we may get into others, but is the concept of the snake's microbiome and how that may give snakes some level of immunity or affect their ability to fight or deal with the disease. So I guess first of all, why don't you, again, sticking with this Bio 101 theme, why don't you give us the Bio 101 on what would a microbiome or a snake's microbiome be, and you can maybe even talk about that relative to their anatomy, say some internal versus external, so on and so forth. But what is a snake's microbiome?
So it depends what microbiome we're talking about, and what we're focusing on in our project is the skin microbiome, which we all have. It's the microbes that live on your skin inherently. They exist there, symbiotically with your body, and they play an important role. They act as kind of a integral first line of defense that will basically complement the immune system. So think about any fungus, bacteria that is present on your skin all the time, pretty much. And it can have a lot of different functions. It can basically, signal to the immune system that there is, something present that shouldn't be there, right? The innate system basically says self or not self, and it can prime the immune system and kind of initiate an attack. It can actually compete for resources. So let's say you get a pathogen on you and you have another fungus or bacteria that has the same need for resources, it'll outcompete it, perhaps. Sometimes there's actual, what they call antimicrobial antagonism, which is what they've found in a lot of amphibian research as being very helpful against BD. And so it is well known, across many, many taxa that we've studied, that there are innate differences between individuals. And it's not always necessarily within, between species can even be different. But we don't fully, especially with the snake skin microbiome, because this is a pretty new area of research, which is kind of, diverged off of all the research that we've done with, BD and the skin microbiome, as well as white nose syndrome with bats. So we're kind of in the earlier stages of understanding how differences in the snake's response to snake fungal disease could be influenced, perhaps, just like amphibians are influenced by the microbiome. And so we're just trying to understand what are the differences. We know there are microbial differences in most, across most taxa. We don't know it very well in snakes yet. We don't know across species that well how different the microbiome is. We just don't have, that, amount of data yet to give us this information that we can kind of extrapolate from. We don't know. Is it, phylogeny based? Does the, all species have the same type of fungus and bacteria on their skin? We don't know. But what about when we look at species differences? Well, maybe there's something else. Perhaps it's their environment that can influence the microbiome. So these are all kind of questions that have gradually come to mind based on, research that's already been done, but there hasn't been that much on snakes quite yet.
Yeah, so it sounds like, and it makes sense because we've been actively working on some of these amphibian-related diseases for much longer. It sounds like there's more work that's been done on this kind of microbiome and disease work with amphibians. And so maybe as kind of a little bit of a framework for what we'll talk about more with snakes, tell me kind of what we know in particular with amphibians, microbiomes in particular, and relative to these diseases. What's been done research wise, and what do we currently know?
There's been a ton of areas of research out there, and obviously I'm not a pro at amphibian microbiome research, but do know that they've done phylogeny explorations. They've looked at environmental correlates. Obviously the effects of particular microbes on pathogens, how they work, species level differences. So I said environment, climate, temperature, all of these things have been explored thoroughly at this point, but we are behind.
So things like different amphibian species, do they have different microbiomes? And then do even say the same amphibian species in different environments, whether that be just like an elevation thing or a disturbance thing or a climate related thing, do they have different biomes? And then how does that subsequently give them some immunity or make them worse off when it comes to dealing with these diseases like chytrid or rhinovirus or one of these? So
exactly. Yep.
Yeah. Okay. Well, let's talk a little bit more about snakes and your research in particular. So as you've already mentioned, there's less known about this topic with snakes than with other groups such as amphibians. So it seems like to me to effectively start to answer this question, it's a complex one because you need to, first of all, have some understanding of snake fungal disease, its distribution, meaning is it present, not present in an individual, in a species. So you need to have the snake fungal disease information. You then need to understand the microbiomes of the snake or the snake species, multiple species, however it's structured. And you need to, connect those, meaning are they related? Is the presence of snake fungal disease related to the microbiomes, or study how snake fungal disease over time impacts species with different microbiomes? How do other factors such as disturbance and landscape affect that? So it's very complicated and it can be done in a number of ways. So what I'm getting at is, why don't you kind of give us an overview of how you designed your dissertation research to get at this very complex question. So I'm almost asking you like the methods of your dissertation.
So I think the big thing that got me, going in this direction is I looked at the research gaps. And I don't want to take anything away from the research that's been done so far. It's great. There's a lot of great information out there, not just on prevalence, but people have been exploring the role of the microbiome, in terms of species level differences, differences across different, types of environmental correlates. But there are like a few gaps. Obviously, we haven't really classified microbial structure across species. We don't really thoroughly understand, is there a species specific microbiome? We just, again, don't have enough of data for that to really say that all species tend to have these specific microbes on their skin. So that we're just kind of lacking, and we need more and more and more studies to look at that. And that's not really a complicated question. It's, do the genetic work and find out what's there. And if enough people get the same answers over and over, that, kind of solidifies things a little better.
So you're looking at the microbiomes. Are you looking at particular snake species, multiple species in your area? How are you designing that?
So yeah, that's one big, and that's where kind of one of the research gaps that I'm kind of aiming at is there have been microbiome studies that have looked across many species. The problem with those research studies is that they have only looked at the bacterial communities on the skin. Then there are studies that have been done looking at both bacterial and fungal communities, but they tend to look at one or two species. And so what I was kind of looking for was a more comprehensive picture where I can look at, kind of a complete picture of the bacterial and fungal, communities across many different taxa. So I wanted it to be kind of all inclusive. That would give us a better picture of, species level differences. And so that was the big question that I wanted to answer. So I knew I needed to look at as many species as possible. And so going into it, I said, I am going to, plan out my sites, which I'll get to in a second, but I will sample any colubrid, non-venomous species that I come across. So obviously I'm not going to be sampling, the copperheads and the timber rattlesnakes. That takes a whole other level of having to not worry about the consequences of picking up a snake. And so I kind of left them out here, although I do want to ultimately include them in research. It just didn't happen in this project. So I said any species I find, I'm going to sample. And that's unfortunate. We all know how hard it is to not only find a snake, but capture a snake. So kind of when I look at the samples I collected, it's a very uneven distribution of species, but I did what I could to represent as many as I could. We got 16 species, but in some cases there were just one or two of them. Okay. But there's enough to look at the picture there. So that's, how I decided on including many species. And I guess your other question was, how I designed my project. My bigger question, because the bigger gap in my mind, is that we're not looking at snake fungal disease or microbiome in terms of or incorporating landscape at all. And I think that's very important, a very important question. They found relationships between landscape modification and wildlife disease among other animals, but we have very limited research on herpetology diseases with relation to the landscape. And currently, there's only been one study so far that has looked at the relationship between ophidiomycosis and landscape, and they only incorporated 2 sites. So I'm hoping that, my incorporation of over, 25 different sites, relatively evenly distributed across natural or forested habitats, agricultural areas, and urban, suburban areas will hopefully give us a better picture of that influence.
Got you. So you're going out and you're sampling snakes across, like you said, was it 16 species? And then for all of those snakes you capture, you're somehow taking samples to look at the microbiomes on the skin. Mm-hmm. And then you, I'm assuming at the same time, you're testing those snakes for the presence of SFD. Mm-hmm. And are you categorizing the snake's health in any way, meaning like some potential impact of SFD, like some characterization of say the amount of lesions and those types of mm-hmm. things, or how do you look at that, or how do you classify that?
Yeah, so my project does have a lot of kind of sub questions going on to make it even more complicated, but that is one thing that we did include in collecting data is, and I'll say this is another gap, that there's really not great standardization in kind of diagnosing or even categorizing the level or severity of clinical signs. And so I wanted to look at that as well and how that correlates with whether or not they actually have the disease and how severe it is. So yes, we used kind of the scoring mechanism where we would look at both the face and the body and give them a score for each from 0, 1, and 2. So 0 being nothing, 1 being we see, a few, crusty scales or a couple little nodules here or there. And then 2, where it looks like it's, either all over the place or affecting, clinical or critical areas like the mouth, cloaca, eyes, things like that. And we gave them those scores, and then we can come up with a total body score, which gives them kind of a clinical categorization.
Gotcha. So for each snake, you have data about the snake, meaning, say, the species, its gender, whatever that might be. You've got whether it has SFD or not. I'm assuming using a genetic test for the disease. And then you have a classification of like the symptoms of SFD, and you have I don't know what you call it, but an analysis or a description of the microbiome of that animal. And then with the landscape component, how do you go about and classify or categorize the landscape for each snake that you capture?
So for that, I'm going to be doing kind of analysis using GIS and a lot of different, land cover databases. And what I'm going to do is kind of create some buffers around sampling sites. And the buffers are going to be, different. There's going to be a small buffer, basically looking at different spatial scales. Basically, daily movement patterns. I do one buffer. Going even larger is like a maximum home range. And then even larger is like max dispersal from the hibernacula. And then within those buffers, I kind of categorize what is there. So I'm going to look at variables such as, urbanization, which I'm going to categorize based on percent impervious surface within that buffer. Agriculture, so I'll look at percent cropland within that buffer, natural or forested area by percent canopy cover, and water abundance based on the amount of like artificial or natural bodies of water that are found within that buffer.
Gotcha. So a lot of our audience won't know what this is, but it sounds like at the end of the day, you're going to have a giant multivariate analysis because you've got multiple dependent and independent variables, and you've got like just with your landscape variables alone, you could have 50s, 100. I know obviously you'll whittle that down to a handful, but yeah. And so let's talk about kind of where you're at, because I know the research is ongoing and it's not complete yet. But let's start with the snake fungal disease. So you have sampled the snakes. What and I believe you have some of the SFD results back, correct? And so what
majority. Yep. Sorry.
I was just going to say. So what did you find SFD wise? In your samples, anything jump out at you? Whether you're passionate about preserving biodiversity, ensuring the survival of threatened species, or simply want to contribute to a healthier planet, there's a role for you. Your support isn't just a contribution, it's an investment in a future where our ecosystems thrive. Go to www.orianne.org to donate or become a member today.
So, yeah. And essentially what we're testing for I wouldn't say it's testing for the disease itself, because just because the pathogen is present doesn't always correlate with, them having the disease state. So what we test for is whether the OO pathogen is present on their skin or not.
Gotcha.
Whether or not it has impacts is, variable. We don't know other than the clinical signs that we wrote down. So we do know what number of samples have OO on them. That's, as far as we can tell, because we're not doing pathological sampling or anything like that. We collected 180 snakes. I'm still about 15 of them. I want to rerun a few times to confirm positive or negative. But we are seeing variable prevalence, and I'm even going back to my notes. It should be up in the, I believe, 20 or so % range is the overall, about a little over 20%. And we did definitely see differences. A big one was between species. And it wasn't a huge surprise, actually, that we saw that our Nerodia species, our water snakes, appear to have the highest prevalence overall, which is not a surprise based on past literature and research that finds that water snakes tend to have a higher prevalence.
Yeah, we found that as well. I mean, I'm sure you've seen those publications, but yeah.
Yeah, for sure. So yeah, it didn't come as a big surprise. I didn't find any big differences between the sexes. Age class didn't come as much of a surprise either. Adults tend to have it more than juveniles, and we believe that's related either to, amount of exposure they've had over time or the shedding frequency. So that wasn't a major surprise. Time of year was, I guess, a little surprising. Sampling year, it varied a little bit, not a whole lot. But yeah, time of year, it wasn't kind of a peak at any one area, except towards the middle of the summer. I think we saw May was our highest prevalence month. Yeah, those were the big ones that stood out. The species county, there were a few counties that popped out, but it's going to take, I'm going to have to do the analysis, the statistical analysis on all this because of our sample sizes being so uneven, between how many we got per county, how many of each species. So I can only give you kind of a big picture overview until I have all of my data and I analyze it. But those are some of the trends that I'm seeing so far.
Gotcha. And I'd say that's pretty typical for most places we've looked for SFD or the actual fungus, not the disease, as you say, but prevalence, pretty high prevalence numbers across a lot of species, but a lot of variation among species. So you've got that component. You've got a lot of the data for that gathered. How about the microbiomes? I'm assuming you've taken the samples, but do you have any? Do you have a handle yet? Have you analyzed the data on microbiomes and on snakes, and what does that look like? And I don't even know how you would characterize the microbiome. So is it, they vary by species, by age, some of these things, but where are you with the microbiome aspect of the project?
So that's relatively early, but it's all just going to come in one big wave, essentially, when I start going on that. Having the DNA extracted was the big part, and running the qPCR was the big part. Now I basically, what's called library prep. I need to basically just amplify certain portions of, universal portions of the bacteria and the fungus, which will then allow me to zoom in on those segments and say, what are the differences between them that tell me that different bacteria or different fungus are present? And so at this point, I've gotten, everything troubleshooted and figured out, what reagents, what primers, the thermocycling parameters, those are all figured out at this point. I just have to do it. And then I have to run the sequencing and find out for each sample what's in there. And so that's going to be done over the next, hopefully 6 months or so. And I can have some good data on that to work with. Yeah.
It's going to be an interesting data set. And you're probably thinking about this, and I may not articulate this well, but it's going to be an interesting data set once you're able to look at the relationship between microbiome and fungal disease. It's going to be interesting interpreting that because, let's just say I'm going to grossly simplify however you would be quantifying microbiome, but I'm going to say high or low. So if you have a snake with a high microbiome, but low snake fungal, it's like a cause and effect thing. You understand what I'm getting at? Would you expect a snake with a high microbiome, or we would expect a high defense to it, to have low amounts of SFD, or would it actually have high because the snake's still alive and it's got high SFD because it has high immunity, or vice versa? It's just interesting how you interpret that. It's going to be pretty interesting.
Right.
I don't know if I articulated that well for the audience, but maybe you can articulate it better than I did as a teacher.
Yeah, probably not, but you're not wrong in saying high. I know what you're saying is you can look at it several ways, and there's diversity is one thing. How many different OTUs or how many different species of bacteria and fungi are present? How diverse is it? And then you can look at, species richness, or how many of each of those are present. And so you can look at, there's one, hypothesis out there that's diversity and invasibility hypothesis that says, if there is a higher diversity of microbes on the skin, then it should be more protective. And I guess it goes back to, more competition and all the other reasons I listed before that the microbiome could be helpful.
But what I'm getting at is, how do you interpret that? Meaning, so let's just say the snake has high diversity. Does a high snake fungal disease measurement, does that indicate that high diversity is good, or does a low snake fungal disease load indicate that's good? How you interpret it? Because that's kind of what I was getting at. It seems like a hard data set to interpret because.
Right.
Yeah.
Yeah. So I see what you're saying for sure. And that is kind of the challenge with this project, because it's not laboratory based. I can't really look at how presence of OO causes a shift in the microbiome over time. But what I'm kind of looking at is, if something has not only high diverse, are they going to have a lower pathogen load or no presence at all? If this hypothesis holds, not really looking at shifts, but what I can do for comparison's sake, as far as understanding the shifts in microbiome go, is look at one, species. If I do see, similar microbiomes across species, I can then compare infected versus non-infected species and look if there is a difference in the microbiome. And so that's kind of my goal with the species that I have a lot of. I'm hoping to see that there are patterns across species, and then that will allow me to look at that differentiation there.
Gotcha. So I'm assuming if you don't have the microbiome data, you're obviously haven't done these landscape analyses yet, but do you have a hypothesis as to, as to how landscape may be affecting microbiomes relative to snake fungal disease impacts?
Yeah. So, like I said, there's very limited research on, herpetofauna and landscape, especially with microbiome. But there have been studies that have found that, environment does shape the inherent microbiome, and that perturbation of it, so disruption of it, can make, if there are changing environmental conditions or urbanization, anything a stressor, it can increase the occurrence of pathogens because it modifies the microbiome. And they've even found that, and I'll give you one study, for example, on amphibians that somebody did. They looked at urbanization and the microbiome on amphibians and found that they had increased, I guess, they call it stochasticity or randomness. So there was a less consistent microbiome across the group, and it was less stable in the presence of urbanization and environmental stressors. So they did show that there is an influence. And so I think that, pollution as well can influence, it can also decrease the immunocompetence of an organism, but it can change the microbiome. If you introduce things like, heavy metals and different chemicals to the environment, I'm sure that's going to alter what's naturally on the skin, especially for something that's slithering around and has their body on the ground the whole time and is covered in whatever's on the ground. And so, I do think that across the landscape, if you're in agriculture, you're going to have those chemicals influencing. If you're in the urban areas, you're going to have different stressors, a completely different microbial reservoir present. And so, I do think that there will be differences, and I do think stressors are going to affect it as well.
Interesting. Well, I'm looking forward to this all coming together. What's kind of your timeline? Where are you at in your PhD program, and when do you kind of anticipate, finishing up your analyses and writing your dissertation and all of that?
I like to work in bursts when I'm not, in the middle of my job. So like during my breaks, my spring break and my fall break and my Christmas break, I'm going to have to be in the lab all the time, trying to get through as much of that molecular work as possible. I'm hoping within the next 6 months to have the microbiome data pretty well ironed out. Obviously, qPCR is basically done, so I have presence, absence data, and so really it's just the next 6 months or so. And then it's time to do all the fun stats and the writing. So probably at least another, 6 months, to a year for that. So a year and a half if I can really, buckle down.
Great. Well, look forward to those results, and maybe we'll have you back on when you complete this and kind of have a better understanding of what's going on in the system. So anyways, well, we'll begin to kind of take this one in for a landing. And I like to have all of my guests tell me their best snake story. So do you have a good snake story for me today?
I don't know. There's been a lot of fun out in the field. I'd say the best part in general has just been taking new people out and experiencing working with snakes. And, I guess this is it. I know the person that I'm going to be talking about momentarily is going to listen to this podcast and laugh that I talked about her. But
feel free to give her name too.
Just in case, I won't give her name, not to embarrass her. Okay. She was a huge help on the project. She came out with me, volunteered, for months to collect all of this. But somehow, over the entirety of all the sampling seasons, managed to evade any snake bite. Despite the fact that we're picking up and swabbing the face and the body, we get to our last sampling season, pick up a water snake. I hand it over to her and finally gets her first bite. And the first thing I do is say, yes. And I high five her just because she's gone the whole way and finally experienced everything there is to experience with sampling snakes.
Was she as excited as you to be bitten by a water snake?
She was, I mean, I was probably more excited, but she also looked at me and said, oh, like that was it. Like, I don't know. She was always so scared and careful and be like, no, you hold it. Oh, it's a racer. No, you hold it. And so it finally happened. And then she became like, oh, this is actually something I shouldn't have been like freaking out about all this time.
Yeah.
But my reaction to it was actually probably the funniest part.
Well, the amazing part of that is that she handled so many snakes, in particular, some species like racers and water snakes, and hadn't had a bite to date. That's crazy.
It was shocking. Yep. It's impressive, I'd say.
Yeah. Great. Well, thank you so much for joining us today on the podcast. I really enjoyed it, and I'm glad we could do a small part to help you with this research.
Thanks so much for having me. I enjoyed talking about my project, and I definitely look forward to giving you some more information in the future.
Great. And I wanted to thank you, the audience, and tell everybody to remember snakes are animals too, and it's a privilege to see one in the wild.